of us, Dr. Rukmini, is going to talk about mechanistic models in novel drug development, going beyond the trial and errors. And she is a chief scientist officer as well as co-founder of Vintage Research. And I knew Rukmini when she returned to India and started this company and making presentations. And I can see that now company has grown quite a lot. No, the spirit, I don't know, but I can. Every time I see her, the spirit is the same. And thanks her for. Yeah, of course, alum of IIT Madras. Yes, yeah, that's important. Yeah. So, without any delay, let me welcome her and please continue with your talk. Thanks.
Thanks, Nirav. So people usually thank the audience for their attention. Today I feel I really owe you gratitude for coming on what would have been a nice day hanging out with your friends watching the match. So thank you. I'll try to make this worth your while. Like Nirav said, my name is Rukmini Kumar. I run a company called Vantage Research. Simply what we are is a computational biology company. And we advise and we consult with pharma companies who are in the process of novel drug development. And I'll talk a little bit about what it is we do and why we are in business. What I'd like to talk today is a little bit of an intro like that and a particular case study. This is in the area of immunotherapy, and this is a really kind of exciting work. Exciting not because it happened quickly, it actually took 5-6 years. So I'll try to kind of summarize the work that took us 5-6 years and maybe not get into a lot of the technical detail, but give you a flavor of where approaches like ours can be useful in novel drug development. So when you think of drug development, there is a kind of ideal way which you may think it happens. We've seen a lot of talks in omics. Somebody spoke about QSAR. There is an idea that you want to identify what is the target that you're manipulating, what is the molecule that will manipulate that target the best, and then you test it in animals, you test it in vitro, and then when you're convinced, you take it to a randomized control trial, which is kind of the gold standard of when you know a drug works or doesn't work. And when all is well and your drug is safe and efficacious, it gets approved by some authority like the FDA, and that's a drug that you can go to the pharmacy and buy. So that's what we think of as sort of an ideal process, but real world is rarely that ideal, and maybe some of us got a peek into that when we were all waiting for the COVID vaccine to come out, and every time there is a trial, we've not recruited enough people, is the PK good, all sorts of questions, we're able to get a ringside view of what happened. So here I'll try to list some of the complexities that happen in the process of drug development. First of all, you assume there is a certain protein or something that you want to manipulate, but whether that manipulating that protein will really lead to an improvement in the disease phenotype, that is something often that we don't have a very strong understanding of. What is the best biomarker to track efficacy? Among the candidates, which is the best chemical which will really manipulate the target? Usually there is a good understanding from, again, structural modeling, but sometimes we need to have leads and backups. And then once we go into a trial, there are a lot of questions. How should the trial be done? What is the dosing? So for instance, in the COVID trials, we heard that one of the vaccines, I think AstraZeneca had a vaccine which they only dosed once, and then whether the efficacy would have been as good as some of the other vaccines which got dosed twice and thrice. So what should the dose be, who should get it, and then once it is out in the real world, what does the evidence really tell us? Is it really supporting what we thought happened in drug development? At the end of it, drug development is expensive, inefficient, and risky process. So this is kind of estimates from a few decades ago almost. Drug development can be very expensive, greater than $1 billion to get one molecule out. It takes a very long time. As many as 100 molecules can be there in the beginning for all of it to funnel out to one successful molecule at the end. So de-risking this process and making it more efficient is a very worthy and valuable goal, and how we can go about doing that is kind of what I'll try to establish in the second half of the talk, especially using this case study. So what it is that we do at Vantage and what modelers can do for drug development is the following. When you think of for instance, let's take the COVID vaccine case again. You're saying, okay, when I inject the vaccine in a population, what are the titers which show up? How many T cells which respond to the COVID antigen that I'm seeing? So all of that is stuff that is happening at the population level, and it's tracked in a trial. But we all know that whatever is happening, there is a lot of perturbation in the biology, which is all happening under the covers. So what all is happening there, and how does that percolate up to what is happening in the clinic? That systematic understanding in a quantitative and integrated way is what a computational approach or computational biology approach can contribute to the process. The other point as well is, this information is increasingly siloed in various compartments. So like I said, if the process takes 10 years, it often takes hundreds of people by the time you design a drug, test it in an animal, test it in a clinic, and finally get it out into a trial. And that expertise can be siloed in protein engineers, translational scientists, biologists, clinical pharmacologists. So how can all of this information be integrated in order to provide a view which takes into account all of these considerations? Okay, so that's kind of the challenge in front of us. In the next few slides, I'll talk about this particular case study. So we'll change gears a little bit, talk about what is new and exciting in the field of cancer immunotherapy, what contribution that we've been able to make, and how did modeling help us make that contribution. So this is work that Vantage did along with our colleagues at Merck Sharp and Dohme. This is a big pharma company located in the US, often abbreviated to MSD. So for those of you that are kind of not familiar with what is happening in immunotherapy, this class of drugs comes with a lot of promise in kind of the war against cancer. And I think the way it really caught the imagination of people is in 2009, this particular immunotherapy drug called anti-PD-1 pembrolizumab, it cured the melanoma of then 90-year-old US President Jimmy Carter, such that usually when you think of melanoma, it's almost always thought of as a death sentence. With all of these extenuating circumstances, it would have clearly been a death sentence. But President Carter is still in hospice, and he was able to beat melanoma in kind of a dramatic manner because of immunotherapy, and that really kind of brought the promise of this drug to everybody. What really is immunotherapy? So when you think of classic cancer cures, most of it is very cytotoxic drugs, right? So you put really what is a poisonous drug on a highly proliferative cell and you get rid of it. There are some targeted therapies as well, which go after particular cell signaling molecules. Immunotherapy works on a different premise. Immunotherapy works on the same premise that the immune system is designed to fight against bacteria, viri, and any xenobiotic, anything that is foreign to the body. So this is a very famous paper on the cancer immunity cycle. And basically, just like bacteria and viri release antigens which will tickle the immune system, cancer cells are also not native cells, and their antigens can instigate the immune system, can lead to the production of T cells which are specifically able to attack these cancer cells, and you can kind of channel or really make the immune system attack the cancer versus relying on cytotoxic agents. That's the promise of cancer immunotherapy. I think there was always a kind of, this promise and this theoretical approach was always there in cancer, but I think only in the past couple of decades have been able to realize that promise in the form of multiple drugs. Okay, so let's drill down a little bit more into what is happening at that bottom layer, especially when it comes to this class of molecules called checkpoint inhibitors. So like I said, you want to tickle the immune system so it recognizes what is a foreign antigen. In order to do that, the immune cell has these sentries, these roving cells which are called antigen presenting cells. They'll pick up all this debris and stuff which they think is foreign and show it to the soldier cell into the immune system, which is the T cell. So they present what is called an antigen. And what is interesting about the immune system is every time you want to jack it up, there is also always a switch to kind of slow it down. This is because an out of control immune system can also lead to something called cytokine storm, and this is how people die in the ICU from all the collateral damage that your T cells can inflict on your own cells as well. So the immune system is always on a knife edge. You want to kind of instigate it, but you also want to bring it down. To that end, there are kind of receptors called checkpoints. As this antigen presenting cell is juicing up the soldier cell, it also has a way to tell it to quiet down. So nearly always you'll see kind of inflammatory cycles. Even when you have a fever, you'll have all of those symptoms and then they'll quiet down basically because of these checkpoints, something which tells the immune system to shush, go down to sleep, we're all done. Now what happens in cancer is it is clever enough to mimic these checkpoints, and so it brings up checkpoints to quiet down any immune cell that comes to attack it. So a couple of key checkpoints are here, CTLA4 and PDL1. These have evolved to quieten the immune system, but the tumor cell has evolved to use that very mechanism to quiet down any immune cell that comes after it. So what checkpoint inhibitors do basically is to stop that connection. They stop that signaling, and they make sure the tumor cell doesn't have a way to signal to the T cell to keep quiet. So those are the cells which come. So then your immune system's power is unleashed and it can go after the cancer. So that's the premise and the promise of what is happening in immune checkpoints. There are several checkpoints. Again, given evolution, there is plenty of redundancy. These are not the only 2 checkpoints. There's a whole slew of checkpoint inhibitors that may be coming, which may be more or less relevant in other cancers. For instance, melanoma may express this checkpoint, but there may be other cancers which express other checkpoints. So this is a very fertile area of research. So now we kind of saw what is happening in the bottommost layer. Let me zoom up, because again, when you're talking about drug development, you're also worried about what's happening in the bottom layer. You're also worried about what is happening in a clinical trial. In a clinical trial, how does a cancer clinical trial look? What are all the things that we care about? The first thing that we care about is what you think of as a classic progression curve. So this is a curve, 100% of the patients come in. Anybody who unfortunately dies or has a relapse of the disease, they fall out of that curve. So this is a typical Kaplan-Meier curve. And the number of patients you have at the end are the patients that you've been able to successfully keep away from progression. So this is the progression-free survival curve. Now, how is this curve plotted, and what really happens when a patient goes into a trial? So I'm going to go into a little bit of detail here. So when a person with melanoma, for instance, goes to the doctor, what really happens is the melanoma usually has metastasized by that time, so they don't have lesions just on their skin, they may have lesions in internal organs like their liver and elsewhere as well. So when they go to the doctor to get treated or go to a trial, the doctor identifies 3 or 4 easily measurable lesions and calls them the target lesions, because they're trying to find out, when I give the drug, how much will the lesion shrink. So that's the target lesion, and what they do is they take the sum of those diameters, watch how those diameters shrink, and then say, you know, if the sum of those longest diameters have shrunk, my drug has been effective in curing this patient's cancer. However, the story is not that simple either. I said the doctor picks up 2-3 lesions which he can track, but there are non-target lesions. People can come with 10-15 lesions, so there are other lesions which they are unable to track. Also, sometimes during the course of the trial, the patient may develop a new lesion. That's called a new metastatic lesion. So you only had lesions in some places, and then you get a lesion in another place. So those are things which can happen as well, and those are also reasons for which the patient may pop out of the trial as a progressor. If you're unable to stop a metastasis, that means your drug has not been effective. Now let's zoom back down. I spoke about a patient having multiple lesions, and I spoke about how the T cells and the tumors can interact. So what is the next thing which can happen is there can be heterogeneity in these lesions. So here on the right, you have what you can think of as a susceptible lesion. It's in this field, we call it a hot tumor. There's tumor cells, but there's also highly infiltrated with immune cells. So all the soldier cells are there. You're really waiting for the checkpoint to remove that break so that it can really attack those cells. So those tumors can be highly susceptible to checkpoint therapy. All the way on the other hand are what we think of as cold tumors. So these tumors have somehow subverted that attraction mechanisms. There are no T cells. Even if you put a checkpoint there, that's really not going to help because there's not enough T cells to kill those lesions. So what we really have to grapple with here is the heterogeneity in the patient lesions. So I told you about patients with different sorts of lesions. If they have multiple lesions and they're all hot, that can be a patient that can really respond to your therapy. If they're all cold, then they're not probably going to respond to therapies. But interestingly, most patients will probably fall in the middle, where they'll have some hot lesions and some cold lesions. And how to kind of grasp all this and figure out how to help the patients is part of what we're trying to do here. I'll just take a couple of minutes because I'll be showing some plots like this just so you can see what happens. And for those of you who are interested in cancer, it's really useful to get to know this stuff because it's a very fertile area, and maybe in the next 10, 20 years, there's going to be a lot of interesting developments here. So when you look at a cancer trial, one way in which they show how the trial has gone on is the following. Over weeks, each of these lines is a single patient. Every patient, in order to normalize, we assume whatever their initial lesions were, we start at 0, and these patients which are green, they've been highly responsive to the trial, and those patients which are gray, they have grown on the trial. So these are the responders and the non-responders, and we normalize it all by starting it at 0. But remember again, here we're talking about the change in the sum of diameters. So we're looking at one aggregate property per patient. Now what we discovered as we were looking into trials, as we were looking into data coming out from our colleagues at Merck, was the following. When you look at one of those lines, it's kind of easy for you to imagine that really what is happening is, yes, I know the patient has multiple lesions, but they all behave politely like this. So that central curve, that aggregate curve, is giving me a good idea about what is happening in the whole patient. But really what we found out, both from bottom-up considerations of how immunogenic each tumor is and top-down considerations of what is happening in a trial, that no, the cold lesions are going to grow, the warm lesions are going to, the hot lesions are going to shrink, and you may leave with an assumption that the patient is doing okay, but really some lesions are responding and some lesions are not responding. So this was our theoretical premise when we started looking into both the biology and the clinical trial. Here's a slightly different chart. This shows what happens to the patient at kind of their best response, right? So it's not over time, this is over patients. These are the patients whose tumors have shrunk by more than 100%. So in all probability, you would have thought these are the great responders, and these patients grew by 80%. So you would have thought, these patients are poor responders. There's standard ways to categorize them using scores called RECIST, and you would kind of visualize a trial like this. For any of you that are interested in cancer trials, a lot of the trials report their results like this in what are called waterfall plots. Now what we found, which was consistent with our understanding, was the following. This is a paper that we published with Merck looking at their trial of pembrolizumab with melanoma. And what was fascinating was, it's a very similar waterfall curve. If you look at these guys, you'll assume these are the lucky responders. If you look at those guys, you'll assume they are the unlucky non-responders because their tumor sum of diameters really doubled. But what we found upon analyzing the data is, yes, you're right, a lot of their lesions responded, but even in the best responders, there were some lesions which grew. And more importantly, even in the worst responders, there are some lesions which responded. So what you think of as an aggregate response really needs to be broken down into like a heterogeneous response to really help what is going on in the patient. And is this merely a theoretical consideration, or does it matter to the patients? So for that, again, we came up with these theoretical curves to kind of see what happens and establish that this was also happening in the data. So here I have example toy example patients, 3 different patients. Here is a patient for whom the doctor picked 4 target lesions, and they're really unlucky because on the drug, all 4 lesions grew. All the lesions that weren't tracked, the non-targets grew, and a new MET also came. So this patient is a non-starter. The drug it was like giving water to them. The drug was completely ineffective on them. So it is very reasonable to call this patient as having progressive disease on the trial. But on the right, you have a patient, all of their target lesions shrank, all of their non-target lesions shrank, but wait, they got a new metastatic lesion. In the current way of evaluating what is happening to this patient is also considered as progressing on this drug, or this patient is also considered as having progressive disease. So these two are very different people, and there are obviously like multiple examples in between, and their treatment needs to be very, very different. So we did another analysis, and what is interesting is we are not definitely the first people to report it. There are plenty of work in the literature on what doctors report as case studies, which they call dissociated response. They're like, when I give immunotherapy, I'm unable to kind of, really classify this patient as a responder or non-responder because of this heterogeneous response. And some doctors, in fact, continue giving immunotherapy drugs to patients even after they have grown a new metastatic lesion. And what we were able to show looking at that data was, if you continue treating after a new metastatic lesions, about maybe 2/3 of those lesions still are somewhat responsive to the drug. So the way you treat a patient after that, after being declared a progressive disease, it's really important that you understand this heterogeneity before you decide how to treat that patient. So this is kind of, there was a lot of work that we did, and this is kind of a summary opinion piece that came out with our collaborator and some senior clinicians in the field. And the crux of the question is, should checkpoint inhibitors be discontinued, supplemented with an orthogonal or site-specific therapy like chemo, angiogenesis inhibition or radiation therapy, or continue as a single agent with close monitoring? So what should we do with these patients and how should we understand this clinical trial data is an extremely important question in this field, and that's sort of being highlighted in this opinion piece here. So, coming back to we're a modeling group, this is a very clinical story so far. So how did our group sort of even get interested in the story, and how are we able to contribute to the story? Well, before we started looking into this sort of data, the core expertise of our group is in developing mathematical models. And these are phenomenological models where there is a description of what we think the biology is. There are growing lesions, the CD8 T cells are able to kill these, and this is the pharmacokinetics of the drug and the pharmacodynamics of the drug, which stops inactivation of these T cells. So these are the sorts of models that we come up with, and very similar to probably a lot of you in the room, we kind of have optimization algorithms, biologically plausible ranges, and we put these two together to be able to reproduce clinical data. But in the process, in the discipline of developing these mathematical models, that's when we figured out that this simple one average tumor per patient or one aggregate response per patient was inadequate to explain the clinical data that we saw. And then we proposed, in fact, a couple of years before any of this work, how should a cancer trial be modeled and what are the considerations for that. So a proposal then was, you often think of what is a single lesion as where the stage is where a lot of these things are happening. But really what you should think of is a virtual patient with multiple lesions, and each of these lesions should have some variability that has been reported by the data. That's not enough. You need to have some kind of stochastic event which predicts how a new metastatic lesion comes, and that's when you'll really be able to reproduce a clinical trial. So that was our recommendation on how to absorb clinical trials into mathematical frameworks that can be then used. The reason we do all of this is not to sort of merely explain what's happening in the data. A big part of what we do is to support our clients as they make decisions. So here's a couple of case studies that we have, and here the question that the client has is very similar to what the clinician was asking before. When I have a patient on pembrolizumab, what is the ideal other therapy that I should combine? So we were able to run predictive simulations on these models, and then we were able to say, you know what, we don't think a combination checkpoint is really going to be effective for all patients. And the logic of what we had is, if you're a hot tumor, if you're very immunogenic, most of the work will be done by your first checkpoint. If you're cold, your first checkpoint is going to be ineffective, your second checkpoint is also going to be ineffective. So there may only be a subset of patients that have warm lesions for whom really checkpoint combination will be useful. So this we were able to say a few years ago when checkpoint combination was a bit of a rage. I feel there may be still cancers in which checkpoint combination is useful, but there is a limit to how useful it can be. This is work in progress, and we are showing it in a conference soon. But basically, the question is, once I know that you've progressed on pembro, which is an immunotherapy, should I add another drug or I switch you from one drug to the other? So the exact question that the clinicians posed, we were able to kind of come up with a quantitative measure of, what is the best strategy. Someday in the future, this may become a very personalized strategy. That is, you're able to kind of biopsy a lesion and figure out what is the exact response another patient may need. But, for now, we came up with kind of a generic clinical trial strategy. So the core of our business is really this. Once we develop our mathematical models, how can we advise our clients who are making these important decisions in drug development, how to go about it and what may be the outcome space they are looking at. So with that, I'll conclude this part of the talk. And all of the work that we did has come out in 4 different papers. The first one is the modeling paper. These 2 are papers which looked at data from Merck and analyzed and showed these results. So when you have clinical data and you're publishing with clinicians, those tend to come in like very high impact journals. And then we also wrote an opinion piece kind of summarizing all of this thinking. So this is kind of the work that we've done. The people that I've circled, they are the ones that have done a lot of the work. There are colleagues at Merck, now moved on from Merck as well as Kannan and Madhav from Vantage were responsible for a lot of this work. Brian Top is our colleague and friend from Merck, and like I said, this work took us 5-6 years, and we really kind of built this thinking piece by piece, and he's really the fulcrum of a lot of this thinking. There's a couple of folks from Vantage here today who also worked on this project, so definitely talk to them if you're interested in more details. So the one thing that I wanted to kind of change gears and talk a little bit about as well is, how does science happen in an applied setting? Like most of the people here are working in academic labs. There's a lot of similarity in what happens in academic labs and in industry. But the one thing is, as we, when you start in your career, you often think that you have one skill. And really the skill that I would say that we have advantage is the ability to develop complex models of disease and be able to simulate them. But as we've grown, what we find is we really become extremely effective in advising people in drug development. And sometimes that involves a very complicated model understanding every aspect of the disease. Sometimes it can be a semi-mechanistic model. Sometimes it can be a model just about the pharmacokinetics. Sometimes we do statistical work, and sometimes we just look at the literature with sort of this multi-scale integrative angle, and we're able to provide good feedback to our clients on how to go ahead in these various stages. So I don't want to go into a lot of detail here, but over the past 10 years, we've been able to kind of impact a lot of projects. At this conference, Manasa is showing some work on a project that we did about installing a device in the eye to address wet AMD. Bispecifics and ADCs are kind of the next frontier when it comes to cancer cure, and we have some interesting posters on them as well, something in autoimmune disease as well. So, and they kind of span the scale, because at this point we're really able to help our clients integrate their thinking quantitatively. The one other point that I'd like to make is a lot of our high impact work really happens when there is interdisciplinary collaboration in a kind of, on steroids. Most of this work, Vantage itself, we have a lot of people with this expertise, biologists, modelers, clinicians, clinical pharmacologists, and really when all of these people come together is when magic happens and your theories get more and more refined. So, here's to more interdisciplinary science. So with that, this is our group. We've been in business for about 10 years now. These are some of the clients that we've published with, and we have a handful of clients that we haven't published with, and this is our very good-looking group. So thank you very much for the invitation. Really enjoyed this talk. Yeah.
Thank you, Rukmini. We have a couple of time for questions. I'll start with student.
Thank you, ma'am. My question is that did you compare for any given location in the patient the heterogeneity across patients?
No. So, rarely is that information, sometimes that information is given to us, but you're right that people think hepatic lesions are kind of harder to attack. Lesions in hard to kind of reach areas are harder to attack, but in this study we didn't do that. Most of our analysis was focused on immunogenicity of the tumor. Yeah.
One more if I may ask. Is there any correlation between the heterogeneity of lesions and the occurrence of a new lesion? Or
that's also a very good question. We haven't found evidence of that in the data as well, but it's entirely possible, and I think Mohit does some work like that, which is basically, when there is more probability of growth or more probability of movement, then the cancer can progress in different ways. We haven't been able to get that degree of data, that degree of resolution in the data.
Yeah, wonderful talk. So one question, so you had these waterfall plots and things like that, right? So the heterogeneity definitely will come from lesions in different regions or whatever. Does the heterogeneity also come from lesions in the same tissue?
Yes, it can be in the same tissue. So the thing is, again, I don't have proper data to, kind of say
No,
that.
I mean, you may be able to subset that, like you go to slide 21 or whatever.
Yeah.
Right? You should be able to subset that to.
Yeah, so the question is, this thing, are we able to say that,
Same lesion.
Same tissue? Sometimes they don't record it. So, again, this is a difficult process, right? The patient's also going through a difficult process. What lesions are biopsyable? Usually, the target lesions are from different tissue. So, there may be data, but it was just not immediately accessible to us. But that's a good line of questioning, which is like close by tumors are likely to respond similar.
Or if you have data about if it hasn't metastasized, it's going to be from
the skin. So in this case, it's melanoma. So I think the metastatic lesion can have different origin even in the same tissue, but it's also, most metastatic lesions are in different tissue, right? So it's migrated from the skin to the liver, skin to lung, wherever else.
The checkpoint inhibitors might lead to cytokine storm. So what about that?
Yes, that is absolutely true. PD-L1, this particular one, pembrolizumab that I'm talking about, has less of a risk. There is another anti-CTLA-4 called ipilimumab, which has, there's a big risk, and there is other classes of drugs called bispecifics. Again, they rely on unleashing the T-cell potential, where CRS is a big risk. So there are 2-3 ways. First, you try and manipulate how low of a dose can you go, so that you don't jump that jump that barrier, and your T cells are still within that. So dose selection is one important way. Another way is dose fractionation. That is, give enough dose in the beginning to exhaust some T cells, so they don't release enough IL-6. The last, or another kind of way, is to give anti-inflammatory treatments at the end, and this people do a lot for ipilimumab. So there's a drug called tocilizumab, which is given for RA, which goes after one of the cytokines. So what people do when patients with cancer go with cytokine storms is to give those sort of anti-inflammatory drugs. The larger point is that sort of risk is not an unreasonable one for a person with terminal cancer to take, and so that's the risk-reward ratio there.
Rukmini, is there any quantification of the patient heterogeneity? So, I mean, clearly they are all metastasized, but to different extents, presumably.
Yeah. So this is 1 SD of just the change in diameter, right? What I would have really liked is if there's data on the T cell concentration for a given patient, so that you're able to say, what we have good data on is what is the T cell distribution across a population. When you take one lesion per patient, then you know that some patients have only 1% T cells, some patients have 10% T cells, so they're hot and cold. But what we don't know is what is that variance per patient. There's just not enough data. So we back calculated that, estimated some 20, and it can be pretty. It's not a very sensitive parameter, but beyond that range it can be sensitive. But I don't know the answer to that question.
Yeah, thank you.
I think we have no more questions. Then thank you, Rukmini.
Thank you.
Excellent
Thank
talk.
you.